Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Sina Sekhenke, and this lesson is brought to you by the Free State Department of Education in collaboration with Phakama Research and Development. Welcome. Right, so today we are going to be focusing on paper 2 topics. I hope today you had a wonderful session with the paper that was sitting in front of you. And all the revision that we did yesterday, I hope it worked for you. Now we're going to look at what is it that we need to look out for in paper 2. All the pointers, all the answers that you need to be able to get into the exam with. So the topics that we'll be covering today are human evolution, genetics, nucleic acids, and meiosis. However, we are going to start with the big topics. So let us check what your exam guideline says. It says for paper 2, you need to study DNA, code of life as your first topic, and then meiosis as your second topic. These are the topics that you did in term 1. Then for term 1 and 2, genetics, remember it was overlapping from term 1 into term 2, genetics and inheritance. This topic now amounts to 48 marks out of your 150, so it's almost 1/3. And then your biggest topic, that is evolution, that was covered in term 3, which amounts to 54 marks of your paper 2. Now, I would like for us today to focus mainly on these two major topics, and then we will just highlight a few things on DNA, the code of life, and meiosis, which you are doing so well on. However, on genetics and evolution, I know some of you are still struggling. So we're going to look at how you are going to answer the questions today. Remember, it's revision, it's about the answers. It's about how you are going to answer the questions. Now, when answering, you must remember that when the markers are marking, there are principles that they are following. So I would like to highlight just a few things that you do not make those mistakes while you are answering. The first one is, if 3 reasons are required and 5 are given, the marker is going to mark the first 3 irrespective of whether all or some are incorrect or correct. So please make sure that when you are asked for 3 reasons, you write 3 of your best. Because if you write 5 and you find that the last 2 are the ones that are correct, and then the first one was the only one that was correct, you are only going to get 1 mark, and then the following 2 are going to be incorrect, and the marker will stop marking there. So we are not going to look at all 5 that are given. So please make sure that you write the correct answers. The second principle that you need to look out for is that one of tabulation. When tabulation is required, but paragraphs are given, candidates will lose marks for not tabulating. So if you can check, when you are asked to tabulate 2 differences, you will see that a mark is not 4, it will be 5. That 5, it factors in 1 mark for the table. So if you do not draw a table with lines that are going across and vertically, it is not a table and you are going to lose that 1 mark. Please do not lose marks unnecessarily. Then, if flow diagrams are given instead of descriptions, if you are asked to describe a process and you write a flow diagram, you are going to lose marks. Then this one, please pay attention to the question because it will be written in capital letters. So please look out for that one. If only the letter is asked for, but only the name is given and vice versa, candidate will lose marks. So if a question says, write the letter and the name of a part that is responsible for breathing, for an example, then you need to be able to write the letter from the diagram and also tell us what that part is in terms of the name. So please make sure that you follow the instructions correctly per question. Right, so we start with our biggest topic, which is evolution. Remember, this topic amounts to 54 marks of your paper 2. Now, this is what the exam guideline says you need to know on evolution. Evidence for evolution, sources of variation, and theories of Lamarck and Darwin. I hope by now you know those theories. punctuated equilibrium, artificial selection, speciation, and human evolution. So it's quite a lot packed in one topic. So let's get right into it. So when you are asked, this is now a question. Remember, we are playing with possible questions that may come per topic. So in this section, you will be asked, or you may be asked, what are the evidence that was used as the line for evidence for evolution? Now, you must be able to mention that biogeography was one of the evidence that was used to support the theory of evolution, to say organisms that are found in different regions, different geographic regions, they share common characteristics which can implicate, or it can mean that they arose from a common ancestor. So biogeography can also give evidence for evolution in that way. The second one, genetics. They did what? They compared the nitrogenous bases and the arrangement of the DNAs of the different species. And when they find the similarities, then they can say that these organisms are related. So using what? Genetics, the nitrogenous basis arrangement. Then homologous structures. Scientists can also use structures that may look alike or that may have a certain plan, like the homologous structures, a certain plan to show that these are much more related, or the arrangement of bones in terms of plan. They may have a different function, however, the plan shows that they may have arose from a common ancestor. So scientists can also use homologous structures as evidence for evolution. Another thing that can be used, the common one, this one you are familiar with, fossil records. Scientists are able to compare the fossils and check the structure and date them, and then they can also tell us about the relatedness of certain species because of the information they get from the fossils when they compare those. So all of these can be used as evidence for evolution. So you need to be able to mention all four. Then moving on. On this one, sources of variation. This question can arise on this topic, evolution, and it can also come in the topic of meiosis. So when they ask you, what are the sources of variation? What brings about variation in the species? You must be able to mention all these sources. There are 5 sources of variation. Now, how do you remember all 5? You are going to use this acronym. You remember the acronym CRRRM. If you don't remember, please take note of this acronym. This is the acronym for sources of variation. Now, let us take a look at the first C. What does the first C tell us? It tells us that crossing over is one source of variation. What is crossing over? Remember, when chromatids of non-sister chromosomes touch at a point called the chiasma here and exchange genetic material, that is crossing over. That process is crossing over. It can bring different, you look at this homologous chromosomes here, they have exchanged genetic material here, meaning you have a different genetic composition for this chromosome and a different composition for this chromosome. Then if this continues to happen, then it will bring variation in all the chromosomes that are going to be formed in one gamete. The second source of variation is random arrangement. Random arrangement of chromosomes also happens where? In meiosis. Look at these chromosomes on this first circle or in this first cell. You see the green ones are aligned this side and then the red ones are aligned this side. But when you come to this one, there are 2 green ones, 1 green one on the left, 2 green ones on the right, and then vice versa with the red ones. You see here, they have changed. How they arrange themselves at the equator is random. It is random. So that random arrangement brings variation because no 2 cells will have the same arrangement. Then in that way, that brings variation. So random arrangement of chromosomes. Please use the correct word, which is arrangement and not assortment. The third one, which is the third R, it is random fertilization. When all these sperm cells are coming to fertilize this ovum, we don't know which sperm is going to fertilize the ovum. The one that swims the fastest is the one that is going to fertilize this ovum. Now, we don't know which one is going to be the fastest, and we also don't know which genetic material it is carrying, because during the process of meiosis, it was genetically different. So all of these sperm cells are genetically different. Therefore, random fertilization will bring about variation. That is your third source of variation. Your fourth source of variation, random mating. If you look at this beetle here, it has a choice of mating with this first one, the lighter one, or the one that has the same shade or it's the same color as its own, or this one, the I don't know what is the color here, but it can shade, it can mate with any of these. So mating with any of these three means it is random, because each and every beetle is carrying its own genetic material. You don't know what type of offspring you're going to have when you mate with this one. So that random mating also brings about variation. So this one is reproduction. Then the last one, which is your M, remember you write CRRR or C, 3 R's and then M. M stands for mutation. That is your last source of variation. During mutation, you know that nitrogenous bases can be replaced or they can be deleted. That brings about variation because it can form a different protein than the one that was supposed to be formed, and that will result in a different or in a various protein that was supposed to be formed. Now, this also brings about variation. Please know your sources of variation. Then, types of variation. There are two types of variation. You have continuous variation and discontinuous variation. So what is continuous variation? Remember, with continuous variation, it is continuous. The characteristics where there is a range of intermediate phenotypes. So for continuous one, it is not absolute. You do not say people are short or tall. Not all short people have the same height, or not all tall people have the same height, but there is an intermediate range. So if you take a look at this example that we have here of the learners in this particular dance class, you see that there are short ones and then there are also tall ones, but not all the short ones have the same height. So the variation here is continuous. It's continuous because there is a wide range of intermediate phenotypes. Those ones that are in the middle is just a wide range between tall and short. There is tall, but tall in a different way. Then skin color, no people have the same skin color. No two people have the same skin color, or to say people are white or black. There are different shades between white and black. So that different shades means there is a range of intermediate phenotypes. So hair color also, some have brown hair, they have blonde hair, they have black hair. So hair color is also an example of continuous variation. Then another type of variation is the discontinuous variation. Now in discontinuous variation, it has characteristics that fall into distinct categories. Now here they are distinct categories, meaning it's absolute. It's either you have that or not. So examples can be blood groups. In blood groups, we have a person that can be blood group A, blood group B, blood group AB, or blood group O. It's either you are this blood group or this one or this one or this one. You cannot have any other blood groups. There are only 4 blood groups. And then fingerprints also. Fingerprints are an example of discontinuous variation because no two people share the same fingerprints. So it means that your fingerprints are your own fingerprints, and that variation is discontinuous because no other person will have the same fingerprints as you. I hope you understand the two types of variation that we have there. Then the exam guideline expects you to know the theories of evolution by Lamarck and Darwin. Right. So we will look at how Darwin would explain evolution according to his theory. He came up with this theory in the 1800s, and he explained evolution using the two laws. When they ask you what are the two laws that Lamarck used to explain his theory of evolution, you are going to say he used the law of use and disuse and the law of inheritance of acquired characteristics. That's how he explained evolution. But let's take a look at these laws. What do they say now? What do the laws say? The first law of use and disuse says that as an organism uses the structure or organ more regularly, this is the use part, it becomes better developed and enlarged. So this is the first part of use. Then the second part says, if an organism does not use a structure or organism frequently, it becomes less developed and or reduced in size and may disappear altogether. Now, this whole part is the part that is explaining the disuse part of the law. So the law of use and disuse, you must explain the use part and also explain the disuse part. What does it mean? Then the second law, he speaks about the inheritance of acquired characteristics. What does he mean by that? He means that characteristics that are developed during the life of an individual, meaning they are acquired because they are developed during the life of an individual. They were not born with them. He says they can be passed on to their offspring. So that is just his law. That is how he explains the theory of evolution. Right. So you must be able to use this principle or this theory. Right. You must be able to use this theory of Lamarck in an example. Sometimes they may ask you to use it in an example or give you a scenario. You must be able to apply it. Now, how would Lamarck explain the evolution of the long neck of the giraffe using his laws? You must be able to apply that. So an example can be in this way. He will say, all giraffes had short necks originally. Please take note of that because this first statement is what makes his theory to be different from Darwin's theory. Because Darwin speaks about all organisms, there was variety, there was variation. But according to Lamarck, he says all giraffes had short necks originally. Then what happened? The giraffe frequently stretched or used their necks to reach for leaves of tall trees. So you see, they kept on reaching from having a short neck. Then causing their necks to become longer. So according to him, just by stretching your neck or just by this giraffe stretching their necks, they are going to be long. Then what's going to happen? The characteristics of long necks acquired in this way was then passed on to the next generation. So they have acquired these characteristics of long necks. Therefore, they are going to pass these characteristics. Please take note of this here. We are talking about a characteristic, not an allele. So a characteristic is something that is external on the outside. Then it is passed on to the next generation. Therefore, it is inherited. So you see, this was the law of use. Because there was no disuse here, the question would not want you to explain the part of the disuse. So you'll only stick to the use. So they used their necks, they stretched their necks and acquired the long necks and passed that characteristic of long necks to the next generation, forming an offspring with longer necks than the generation before. So according to him, this is how he would explain using his laws. What are the things that you need to remember when answering Lamarck's theory? First, let us check this terminology here. What is an inherited characteristic An inherited characteristic is a characteristic that an offspring is born with, because we have now inherited it. Having been inherited from one of the parents, a characteristic is controlled by a gene. So this is what we know, that when you inherit something, it must be controlled by a gene, and you inherit it from the parents. Then, what is an acquired characteristic? An acquired characteristic is a characteristic that an offspring is not born with. So you are not born with this one, but it is going to develop through the course of a lifetime of an organism. So whatever that you do while you are living, whatever that you are acquiring, it is an acquired characteristic. You acquire it throughout your lifetime. And this characteristic is not controlled by a gene, because you are already born with your full set of genes, but hence you are acquiring what a characteristic in your or during your lifetime. There are guiding questions that can help you in answering any question using Lamarck's theory. What are those guiding questions? First, you must ask yourself, what was the original characteristic? Like we said with the giraffes, all giraffes had short necks. You must identify from any scenario that you are given what was the original characteristic. And that original characteristic, you must say that all of those organisms had that original characteristic. And then what was the challenge? What made them to want to change? You must identify that. Food, they could no longer reach food on higher trees, so they had to stretch their necks. So what did the organisms do, or what characteristics was then acquired? They stretched their necks and acquired a characteristic of long necks. What was the result? All giraffes now had long necks. Then what happened to this acquired characteristic? This acquired characteristics was passed on to the next generation. Then what was the result of this? Then you end up having a whole generation of giraffes with long necks than the generation before. So all these questions can help you to get full marks when you are applying Lamarck's theory. I hope you remember that. Now, because we are scientists, we know very well that Lamarck's theory is rejected. Now, a question can ask you that, why was Lamarck's theory rejected? You are going to give these reasons as they are. These are the answers to that question. The reasons why Lamarck's theory was rejected is because there is no evidence that acquired characteristics are inherited by offspring. So there is no evidence to say that this was an acquired characteristic and we see it in the offspring. It means there is evidence that this was acquired. So there is no evidence to show that. Then the second reason, it is because organisms did not evolve because they were determined to change, but changes took place randomly due to mutations. So there's no way that organisms can change or can evolve because they were determined to. So we cannot accept this theory, because organisms do not evolve because they want to. Then an organism cannot change its DNA. We know. For you to have a physical characteristic or a phenotype, it must be informed by a genes or the DNA, and an organism cannot change its DNA. If your DNA, you are born with short neck, you will have your short neck until, because that phenotype is controlled by a gene. Then according to Mendel, it is the phenotype that determines it is the genotype that determines the phenotype, not the other way around. So you cannot say an acquired characteristics will inform what the genes of the next offspring or of the next generation will be. So these are the reasons why Lamarck's theory was rejected. Please take note of this, because the question will ask you, if you are applying Lamarck's theory, then there might be a sub-question that is asking you, why was his theory rejected? These are the reasons that you are going to mention. Then we come to theories of evolution by Darwin. Darwin wrote a book, The Origin of Species by Means of Natural Selection. He published this book in 1859. Now, his theory of natural selection has two main points. The first main point, he speaks about species were not created in their present form, but they evolved from the ancestral species. That is the whale evolution. So he used this whale evolution as an example. So what is this statement saying? Is that what you see today is not necessarily how an organism started. It may have evolved from the species that lived before. That is what Darwin is saying. And then the proposed mechanism for evolution that he used is evolution by natural selection. This is where the environment is going to be the selector of a suitable characteristic or a favorable characteristic, and therefore that characteristics will be passed to the offspring, and therefore having a greater generation of offspring that have that favorable characteristic. So he speaks of natural selection. So what does it say? You must know how he explains the natural selection. You may be asked to apply it in a scenario, or you may be asked to write it as generic as it is. Please use your exam guideline to check how this theory is explained, how you should explain this theory. Now, Darwin's theory of evolution by natural selection states that there is a great deal of variation amongst the offspring. You see, that is the first difference from Lamarck. Lamarck was talking about all of the organisms having the same characteristics initially. But Darwin starts by saying there's a great deal of variation, meaning you will find giraffes, if he's using the giraffes, you'll find the giraffes with short necks and the giraffes with middle necks and the giraffes with long necks. So there's a variation amongst the offspring. Then he explains this variation. You must tell us, what is that variation? Some have favorable characteristics and some do not. Then, when there is a change in the environmental conditions, or if there is competition, so now you say everything was okay, there was variation, some had favorable for that particular environment, some did not. Then that environment changes. Now you are disturbing the situation. That environmental condition, or there are different environmental conditions, or when there is competition, you can just imagine what's going to happen. The organisms with characteristics which make them more suited will survive, whilst the organisms with unfavorable characteristics will make them less suited and they will die. So that is a scenario that everyone knows, that if the environment does not favor you, you are going to die. But if the environment favors you, you are going to survive. Then, do you still need to talk about those ones that died? No. You continue with the ones that survive. You tell us, the organisms that survive are going to reproduce. And when they reproduce, remember, genes are involved. They pass the allele. You see, now we are no longer talking about the characteristic. With Darwin, we talk about an allele. They pass an allele for the favorable characteristic to their offspring because they had the favorable one. They can only pass the favorable one because they survived. Then the next generation will therefore have a higher proportion of individuals with the favorable characteristics. Please conclude your statement as to explain how organisms evolved by natural selection. This last statement will be your concluding statement, and it is also very important. Please know the differences between the two, how Darwin would explain it, how Lamarck would also explain it. Then you come to the most recent theory of evolution by Eldridge and Gould in 1972. Now, 1972 is like the most recent time. Punctuated equilibrium is what they came up with. They said evolution took place in this way. There was the punctuated equilibrium speaks to the speed at which evolution takes place. Meaning, there was a time when there was no evolution, and then followed by a period of rapid change. So they are explaining evolution in that way. To say it's the speed at which evolution takes place. And this speed, it involves long periods of time where the species undergo little or do not change at all. And that will be equilibrium, from the punctuated equilibrium. So the equilibrium stage will be that one where there is no change or there's just a little change. Then it alternates with, or it is punctuated, short periods of time where rapid changes occur through natural selection. So they are not disputing natural selection. They are focusing mainly on time, the speed at which this evolution is taking place. So they will say that there will be long periods where there's no change, followed by short periods of rapid change, of fast change by natural selection, during which new species may form in a short period of time. So theirs is mostly concerned with time and speed. These diagrams here are showing you gradualism. This is natural selection as it happens gradually. It is slow, and you see progress. With gradualism, you see progress from the previous species to the next species. There is little change, and the species will also deviate or divert to other species and form other species, but you can still trace the change. It's like you see it coming gradually. However, with punctuated equilibrium, rapid change occurs and it is followed by long periods of no change. So you see from this ancestor species, you see there was no change for a long time, and then rapidly, when the graph changes or when this diagram changes, there's a sharp curve to the right here. It shows now this was a rapid evolutionary change, because you did not see this growing gradually from this ancestor species to this species. Then, and so it goes. From here, you see there's equilibrium, no change. And then here, when it turns sharply or sharp, you see that will represent your rapid change. So that is how you identify punctuated equilibrium and gradualism. Then you also need to know artificial selection. What is artificial selection? Artificial selection is the intentional reproduction of individuals in a population that have desirable traits. So the human here is the selective force. You select the trait that you want to reproduce, then you just reproduce that to give you those desirable traits. So it happens mostly in agriculture. You see the crops, the cows. You can have cows of a characteristic that you want that gives you more milk or more meat. Chickens that are more muscular, they give you more meat. So different types of the what is it, the cabbages. You can have, you can just play with whatever characteristics. As a human being, you intentionally reproduce those individuals of that characteristics that you desire. So that is your artificial selection. Then this diagram shows you the terminology that is very important for the next topic that we are going to deal with, which is speciation. So from grade 10, you must remember how you define the term species. For you to call an organism a species, or a living thing a species, it must have three things in it. So what is a species? Species are organisms that have the same characteristics, one, that are capable of random interbreeding, two, and produce a fertile offspring. So for them to be called organisms of the same species, they must have the same characteristics, they must be able to interbreed randomly, and they must also be able to produce a fertile offspring. Then you say these are organisms of the same species. Then from there, what is a population? You see here, this was an organism or a species. Then you come to the next column, this is your population. What is a population? Individuals of the same species. You see, these are the same species of these organisms. You see, individuals of the same species living in the same area, very important, occupying the same habitat, that can randomly interbreed. Then what you are adding here is the part that they are living in the same area, then they form part of a population. Then, what is speciation? Speciation is the evolutionary process by which populations evolve to become distinct species. So they are forming new species. Right. Then what is geographical isolation and reproductive isolation? You are going to be able to identify this in your statement and also tell us what is separating or what is isolating organisms reproductively. So these are mechanisms to isolate the gene pool of the species, resulting with the formation of new species. So that will be your geographical barrier, isolating these organisms from being able to interbreed and produce a new gene pool. Then reproductive isolation is that when you now have new species, still they are isolated because of certain number of things that make them not to be able to interbreed. Then we will look at those at a later stage. Then what is speciation? Speciation, you will also be expected to define speciation using an example. They can give you an example and ask you, how do you think these organisms came about using speciation theory? Now let's check. Speciation is the evolutionary process by which populations evolve to become distinct species. So how does it happen? You must be able to describe this process step by step. Speciation through geographic isolation is that happens in this way. First, a population is split into two by a geographic barrier. So, split into two. What is split? Please do not say species. It's a population. Population is split. So, if you take a look at this ancestral population here of these rabbits, you say this was the population, they were living in the same habitat. Then they were separated. You see here, they were separated by something that looks like water or what, you see? They were separated. But what do you say? It's a population that is separated, split into two, and it was separated by a geographical barrier. In your answer, check from the scenario that you are given, what is the geographical barrier? A geographical barrier can be a river, a sea, a mountain, or a lake. And there can be a compulsory mark here. If you did not mention that particular geographical barrier, you may lose that one mark. Please make sure that you mention it if it is mentioned in the question. Then, since they split into two, what is going to happen? There is no gene flow between the two populations. See? Now, these populations are exposed to different environmental conditions because the conditions on this side cannot be the same as the condition on the other side. Then, each population undergoes natural selection independently. Now, you cannot say that there is natural selection and then you explain it. Don't explain it. Just say that they undergo natural selection independently. We will understand what you're saying to say those ones with the favorable characteristics survived. What you don't need to mention that. Just say that this population underwent natural selection independently. Then genotypically, one mark. Phenotypically, another mark. They are different. Why? Because the environment was the selective force. Then, even if they were to mix, that is very important. One mark for them. They will not be able to interbreed. Why? Because the two populations are now different species. Do you remember the definition of a species? For them to be able to interbreed, for them to be able to be the same species, they must be able to interbreed and produce a fertile offspring. So this is how you explain your speciation. Very important. Take a look at the flow of the answer, then your answer will be logically correct. This is an example of the species. This was from a question paper in 2021. It shows you the present day distribution of the three closely related species of the dog family. Now, the coyote, the jackal, and the dingo. is shown on the world map below. Now you see the jackal is something, it's different from the coyote, is different from the dingo, but you can tell that these somehow were related at some stage. They share a common ancestor, but because of that separation, they became different species based on their environmental needs where they were situated. Right. Then we come to the mechanisms of reproductive isolation. Once these species have become new species and they are not able to interbreed, what is it that is separating them from being able to interbreed? You must mention this as they appear. First, breeding at different times of the year. This can be something that is isolating organisms to reproduce, because if the other one breeds during July, August, September, October, and this butterfly here, the second one, breeds during March, April, May, June, the times are not the same. So it means they will not be able to interbreed. Therefore, breeding at different times of the year is reproductively isolating the two species of butterflies. Then, species-specific courtship behavior. Remember the intricate dances, the peacock, you remember all those. Now, if the species are not in tune with the behavior that tells them that they want to mate, that can also isolate them and make them not to be able to understand that this one is trying to interbreed with me, because the courtship behavior now is now different. They've learned a new behavior trying to court the other one. Then plant adaptation to different pollinators. You see, some plants are adapted to be pollinated by bees, some are adapted to be pollinated by birds, some by wind. So it will depend on which one. So, but if you were adapted to be pollinated by bees, and then the wind comes or the bird comes, it will not be able to reach the part where the bee will reach to be able to interbreed that particular spores or whatever seeds. Infertile offspring. When you take a donkey and a horse, or a horse and a donkey, you get a mule. Now, a mule is infertile. A mule is infertile. The only way for you to get a mule is if you interbreed a donkey and a horse. You cannot take a mule and produce and interbreed it with another mule and produce another mule. I hope you get it. So infertile offspring can stop or can isolate organisms to reproduce. Also, prevention of fertilization. Here, prevention of fertilization can happen because look at the sizes, look at the different copulation organs, they may be different. So that can also prevent fertilization. So these are the mechanisms that can isolate organisms from being able to interbreed once they become new species. Please know them off by heart. A question can arise asking you to mention them. Then, evolution in present times. Evolution is continuing even today, and we saw that during the COVID-19 that evolution was happening because the virus was changing rapidly, having different names from different strains, you see. So this evolution can be seen in organisms or that are reproducing fast or rapidly reproducing. Mostly it's those small organisms like your bacteria or your viruses. So here, let us take a look at this diagram here, an example. You see that there is antibiotics. You see the antibiotics here? Antibiotics are used to kill bacteria or pathogenic bacteria. Then you have a population of mainly susceptible bacteria. Now look at this. This bacteria is going to be killed by this antibiotic. But within this bacteria, you see the two red dots there. That is the resistant bacteria, meaning this bacteria will not be killed by this particular antibiotic. It will not be able to kill it. So what's going to happen? Over time, the yellow bacteria is going to die because it is susceptible to this antibiotic. It can be killed. However, look at what is happening with the resistant antibiotic. First, there were 2, and then now there are many. They are growing. The red one is now growing, meaning it is evolving more and more rapidly to become stronger and stronger until we have a bacteria that is resistant to this antibiotic. This antibiotic will not work on this red resistant bacteria. Therefore, it will keep on evolving and becoming wiser and wiser until you develop a new antibiotic that will kill this particular strain. So you can see that also with the HIV virus. So the small and rapidly producing organisms are the one that can show us the evolution in present times. Right. Then we come to the next section of this topic, which is human evolution. With human evolution, there are a few things that you need to know for you to collect those marks. You must be able to interpret phylogenetic trees. Example, then you must be able to know who is the common ancestor. Are you able to identify that? Who is extinct? When did the species on this diagram originate? And how long did they live for? And when did they become extinct, etc.? Are you able to read that information from a phylogenetic tree? You must be able to know the similarities between the African apes and the humans, the ones that we share with them. Those are the similarities. You must also be able to tell us about the differences between the African apes and the humans. You must know the diagrams, the skull, bipedalism of the foramen magnum, the spinal cord, the pelvis, etc. They can give you the dentition, the teeth. You must be able to differentiate depending on the diagram that you are given. Then fossils, genetic and cultural evidence that was used for evidence for evolution or for evidence as evidence for human evolution. Then you must know the fossils that were found in South Africa as well as the scientists who found them, make a table. Then you must know the out of Africa hypothesis. So let's get on with it. What is a phylogenetic tree? I know that a question will ask you, what type of a diagram is this? Some of you like to confuse the pedigree diagram with a phylogenetic tree. Please make sure that you know what is a phylogenetic tree. A phylogenetic tree is a schematic form that shows evolutionary relationship. So phylogenetic, remember that one is the one of evolution. Evolutionary relationships within a set of organisms or groups of organisms. Then phyllo in phylogenetic tree stands for the phylum or the phylum group of that particular organism. Genetic, it is from the genes or the relationships between the genes. Then it should not be confused with pedigree. So this is what I was talking about. You cannot confuse this one with a pedigree. Then an information that you're supposed to gather from a phylogenetic tree is that you must be able to identify a common ancestor who is extinct, when did the species on the diagram originate, how long did they live for, and when did they become extinct. Are you able to read that information? Here is an example of a phylogenetic tree. Right. This phylogenetic tree, you must be able to identify the first thing. Look here, it says time in million years ago. 5 will be your oldest time. It will be far in the past. So the 5 will represent the past. And then the 0 will represent the present time. Once you have identified this, you will be able to answer questions. Now, this phylogenetic tree can come in different directions. You can see the one that is facing downwards, one that is facing upwards, or it can face to the right, or it can face to the left. What you need to identify is where is the present and where is the past, so that you can be able to answer the question that when they ask you what is the common ancestor, the common ancestor will be here. When they ask you for the common ancestor for all hominids, you are going to write the species that is here. This is your common ancestor for all of them. But if they ask you for the common ancestor of the Homo habilis and the robustus, your common ancestor will be here. Then it will be your Australopithecus afarensis. You see, what comes before the branching is your common ancestor. Then the branching shows the descendant. What descended from the common ancestor. So this branch will show you these are the descendants of this particular common ancestor. Then when a line becomes or stops like this in the middle, you must know that this means that the species has become extinct. Then when it branches in this way and still continues to the present, it shows that there was speciation that took place there. So this is all the information that you can gather. Then you will be able to answer the questions that when did this organism become extinct? You'll be able to tell us in terms of millions of years ago. Then you can also tell us when did the Homo habilis originate, or what time did they start appearing. Then you'll be able to use this information there on your phylogenetic tree. So I hope you are able to read the phylogenetic tree no matter how it appears. Then another thing that you're expected to know is to know the similarities between the Homo sapiens and other primates. So the similarities, they will not ask you all 10 or 12 of them just for you to list them. It can appear in a question where they say there was other questions leading before that one, and then you are asked to identify what is it that we share. When they say we share with other primates or with African apes, then you must know they are talking about their similarities. So let us take a look at all those similarities that we share with other primates. The first one, when you look at the head of this particular diagram here, it shows you the head. The head represents what? A large brain or large skull compared to their body mass. So all primates have a large brain or skull compared to their body mass. Number two, remember this. Use your body. Remember, use your body. You have all the answers within you while you are seated in the exam room. Start from the head, go down. You will get all your answers. There are more similarities here, but I'm sure you'll be asked for 4 or 5. They cannot ask you more than that. They cannot ask you all 12. So, but you must score. Make sure that you score on this question. Eyes. When you come to number 2, eyes, it shows that the eyes are in front, or they have binocular vision or stereoscopic vision. All primates have that. 3, on the upper arm here, shows you what? They all have freely rotating arms. You can even try to rotate your arm there in the exam, but without disturbing other candidates. Then number 4, longer upper arms. 5, at your elbow. Elbow joints allowing the rotation of the forearm. Then 6, this one, anyone can get this one. 5 fingers. All primates have 5 fingers. Then 7, an opposable thumb, meaning a thumb that is working in the opposite direction with the rest of the fingers. Then 8, flat nails instead of claws, or they have bare fingertips. You can write it like that. That is number 8. 9, 2 mammary glands in front. Then going back to the eyes. Eyes, you can also say that they have eyes with cones for color vision. Then sexual dimorphism, also there represented in the eyes. Sexual dimorphism means there's a distinct differences between the male and the female. So these are all the characteristics that we share. Please just list them. This one is a level 1 question. You can get marks there freely. Another question that can arise are the differences. With the differences, expect to be given a diagram. It can be diagrams of the skulls, can be diagrams of the palates, diagrams of the pelvis, diagrams of the spinal cord. So you must be able to differentiate between the humans and the African apes, looking at the different diagrams. So first, the first difference is that humans show bipedalism, whereas the African apes show quadrupedalism. Now, bipedalism is brought about what? The foramen magnum is in a forward position. So this is on the skull. You are not going to say the foramen magnum is below the skull. The correct terminology to use there, you say the foramen magnum is in a forward position. Why are we saying forward? It is because we are looking at the evolution of where it started with other primates that came before the humans. Then it moved towards the front, but we say it moved in a more forward position. And then with the apes, because they show quadrupedalism, the foramen magnum is towards the back of the skull. So this one is towards the back of the skull. Then, leading to what? Look at bipedalism. The spinal, the vertebral column or the spine, not the spinal cord. When you say the spinal cord, you are talking about something else. You'd rather say the spine or the vertebral column. It is S-shaped. Whereas with the African ape, the spine is C-shaped. Then what is the pelvic? Please use the correct words that are used here. The pelvis of the humans is shorter and wider. So you don't say it is, you don't use another synonym for short that may be meaning something else. Please just use short, and you don't say small. Yes, because some of you are tempted to use the word small and maybe wider to say small and big. You see, now you're contradicting yourself. So use the correct terminology. The correct terminology to use there is short and wider. That is the pelvis of the humans. And then the pelvis of the African apes is long. The one that is long and narrow is the one of the African apes. Then let us look at the cranium size. The cranium size is this part of the skull, not the whole skull, just the cranium of the skull is larger, whereas the cranium size of the African ape is small. Remember, the cranium size is that type of the top of the skull, the cranium. Then the palate shape. The palate is the arrangement of the teeth. So the palate shape for the humans is round. It has round jaws. Then the palate of the African ape, it is a rectangular jaw. So I think this is where you can also score marks easily. Then the dentition, how are the teeth arranged within the humans? Humans do not have gaps in between their teeth, and they have small canines. Whereas the African apes have gaps between their teeth, which are called the diastema. And they have larger canines. And there's a reason behind that. It's because they had to tear raw meat, whereas we eat our meat soft and cooked. Then non-prognathous, the face of the humans are flat. Even if you take a ruler, you are going to draw a straight line across on the face. However, with the African apes, the faces have a slopy face. So if you take a ruler, you are going to end up with a line like this in the face of the African ape. So they have a slopy face, not slopy, like it has a slope. Then the cranial and the brow ridges. Now the cranial ridge is that ridge on the cranium. And then the brow ridges are those bridges on top of the eye sockets there. So with the humans, they are absent. However, with the African apes, there is a cranium ridge and also the brow ridges. Right. So those are the differences. Get those full marks. So here you are going to study the diagrams of the skull, the pelvic girdles, and the vertebral column. You see all of those to give us these differences. Right, so want to go next? Right, lines of evidence for human evolution. Remember, we had lines evidence supporting evolution as a whole, but now for human evolution specifically, what evidence did they use to support this evolution, or to support this theory? Fossils evidence, that is what they use. and transitional species. Transitional species are those species that have characteristics of common to both the ancestor species and the descendant species that follows. So this one is in the middle. So scientists are able to study fossils and date them, and they are able to put the fossils of organisms in between to say, but this fossil have the characteristics of the previous species and the characteristics of the next species. Therefore, it is a transitional fossil and it gives information to provide evidence for human evolution. Then also, genetic evidence, mitochondrial DNA. With mitochondrial DNA, it is used to study the ancestry of females. Remember, the females are the only ones that can pass their mitochondrial DNA to their offspring because their ovum is fertilized as it is with its own nucleus, with its own mitochondrion, whereas the mitochondrion of the sperm cell is cut off during fertilization because only the nucleus of the sperm cell enters the ovum. So that is why the females are the ones that we are able to trace the mitochondrial DNA from. So we study also the mutation that take place in mitochondrial DNA, then to prove that this is how evolution takes place, or to prove the relatedness of the humans or human evolution. Yes. Then cultural evidence, tool making. Scientists are able to study the development and the use of tools which separated humans from other hominids. So as time went on, you see there are different tools that are progressing, then you can tell that the species lived during this particular time and they use these particular types of tools, and therefore they are relating, they are providing some evidence for human evolution. Right. Then what you need to know is the fossils that are found in South Africa. Know the scientists that found them. This one can appear in terminology. It can also appear in multiple choice questions. So make sure that you are ready for this type of question. They may ask you about this. Let us look at the first species. The first species that were found were Australopithecus africanus. So there are 3 examples of Australopithecus africanus that were found in South Africa. The first one is Dawnchild, which lived when 2.3 million years ago. Then it was found where? In the village of Dawn. You see, Dawnchild, it was named after that village where it was found in South Africa. Then it was found by Raymond Dutt in 1924. That's a long time ago. Another example of Australopithecus africanus that was found in South Africa is Mrs. Pless, whereas there is also a debate now to say it was a Mr. or it was a Mrs., but just write Mrs. Pless for now, which is widely acceptable. It lived on the earth about 2.5 million years ago, and the fossils were found in the Sterkfontein caves in South Africa by Robert Bloom in 1947. Then the last example of Australopithecus africanus, you can be asked to give the 3 examples of the Australopithecus africanus fossils that were found in South Africa. You are going to mention Dawnchild, Mrs. Pless, and Littlefoot. Littlefoot lived around 3.9 to 4.2 million years ago. It was also found, the fossil was also found in the Sterkfontein caves by Ron Clark in 1994. Then another species that was found in South Africa is Australopithecus sediba. An example of Australopithecus sediba is Karabo, which lived about 1.95 million years ago. It was found near the Malapa caves in South Africa. Then it was found by Brett Iloff and Lee Berger in 2008. So this is just a memory. You need to just recall this information as it is. And then the other species is Homo naledi, which means star. It lived around 335,000 years ago to 236,000 years ago. You see, this one is not million years ago, so it is the most recent. Then it was found in the cradle of humankind in South Africa by Professor Lee Berger in 2015. Then the Homo sapiens, the species that are still thriving even today, the flores-backed men lived around 200,000 years ago. You see, not million years ago, just years ago. It was found where in the Free State, Makapansgat in Limpopo border, cave in KZN, the Blombos cave in Western Cape by Professor T.F. Dreyer in 1932. So these are the fossils that you need to know and the species that are found in South Africa. The last part that you need to know is out of Africa hypothesis. Out of Africa hypothesis shows that what is the evidence for out of Africa hypothesis? What does it mean? What does the out of Africa hypothesis mean? It means all hominid species originated in Africa and moved to different parts of the world. So you see everything, you see there's on this map, the red is in Africa. And then you see all the arrows moving to different parts of the world. So that is what is meant by out of Africa hypothesis. But you need to be able to mention what evidence is there to support the out of Africa hypothesis. Fossil evidence was there to support this theory. What fossil evidence was there? It showed that Ardipithecus fossils were found in Africa only. If you can remember the Ardipithecus are the oldest ancestor of the hominid species. So if you find the eldest ancestor only in Africa, that can tell you that from the ancestor, everything originated there in Africa and therefore spread to all other parts of the world. This is the first reason or the first evidence. Australopithecus fossils are found in Africa only. So Australopithecus came after Ardipithecus. So it's still the oldest ancestor. So if Australopithecus fossils are found in Africa only and nowhere else in the world, that can tell you that it means the hominid species originated where? In Africa. This is now out of Africa hypothesis being supported by this statement. Then Homo habilis is found in Africa only. Homo habilis, we are still going back further. Homo habilis is found in Africa only. So as they come from Ardipithecus to Australopithecus to Homo habilis, they are found in Africa only. So those three are the eldest, they are the oldest. And if they are found in Africa only, that can support that hominids originated where? In Africa. Then also, the oldest fossil of Homo erectus is found in Africa. What does this mean? Fine, Homo erectus was found in other parts of the world. However, the eldest fossil or the oldest fossil of Homo erectus was found where? In Africa only. So still supporting what? That origination from Africa. Then genetic evidence that was used was the mitochondrial DNA. The analysis of the mitochondrial DNA traces the oldest female ancestor to East Africa. So that genetic evidence also still supports what? The out of Africa hypothesis. These reasons, you mention them as they are. You do not change them. You do not try to interpret. Just mention them as they are because they are supporting, they are giving you now, this is the evidence that was used to support the out of Africa hypothesis. Right. Then we are done with evolution. That was quite a big topic. I hope you will be able to collect those marks, 54 marks of your paper 2. Then we come to the next big or the second biggest topic of paper 2, which is genetics and inheritance. This topic amounts to 48 marks of your paper 2. 48 marks is still a lot of marks. So I will need you to collect these marks. This topic has a lot of terminology that we find that learners are confusing the terminology. Go back and check the terminology that is written here. These concepts are mostly confused by you candidates and you may use them interchangeably and they become wrong in your sentence or in the construction of your answer. So go check the word chromatin versus a chromosome, what those are, genes versus alleles, dominant allele versus recessive allele, phenotype versus genotype, and homozygous versus heterozygous. So those are the terminologies that you need to know when to use which one. Then we are going to talk about them in the next section. So with genetics, we know that we have the grandfather of genetics, which is Gregor Mendel. Gregor Mendel is the one that came up with these laws as he studied genetics. Then he has the principles that explains how organisms inherit the characteristics. So the first law that he used is the law of inheritance. He says the law or the principle of segregation. So he speaks about the law of segregation. No, you will be asked about how Gregor Mendel explains the laws of inheritance. So it's the law of segregation where the gametes are going to be segregated. And then the Mendel's second law of inheritance is the law of dominance, where the dominant allele will cover or will be dominant over the recessive one. So he speaks about the law of dominance. And then his third law of inheritance is the law of independent assortment. So those gametes are going to assort them independently, irrespective of where the other one is. So he studied this genetics and came up with these laws. So we see these laws when we are doing a genetic cross. We practice all three laws. You will see them in practice, but be able to mention them. Right. So a genetic cross. Here I'm going to plead with you. When you are given a genetic cross and you don't know the alleles and you don't know how to cross, do not pass that question. There are 3 marks that you can collect for free. Please make sure that you write something for you to get those 3 marks. A genetic cross is mostly asked out of 6. So you can get, 3 marks out of this genetic cross. I will show you how. So this is the layout or the format of a genetic cross. First, the visible trait that is the phenotype, example, the tallness or the shortness. In the question, you will always be told that when you cross a tall tree with a short tree, or with a mother with blue eyes with a father with brown eyes, you see, they are explaining the physical characteristic, which is the phenotype. You take that information as it is. You come and you put it here. You put it here where there are question marks. They will tell you when you cross a mother with brown eyes or a father with blue eyes, you write it there as you take it from the question. Already you have scored 1 mark. As simple as that. You don't have to think about it. You write it as it is from the question. Then you come to the genotype. The genotype is the genetic makeup of an individual. So what gives that person the blue-eyed colors are the alleles. So you are going to write out the alleles or the genes that gave that person the blue color. This is where you are going to look at the colors, the letters. Use the letters and look at the situation where what is said in the statement. When they say a heterozygous for blue color, you know that there is one allele that must be dominant and one that must be recessive. It will be like this. Small t, big T, small t. But when they say it is homozygous for blue color, you will use 2. Homo means the same. So you will use 2 dominant depending on whether that color is controlled by a dominant gene or a recessive gene. You will get this information from the statement, and therefore sometimes you can have recessive for both. The alleles can come in a recessive format like this. So here, this is most of you where you get the challenges. You cannot determine the letters. You don't know how to use homozygous or heterozygous. Then you lose the marks for writing the correct genotype. More especially if you were not given the letter to say, using the letter B, do a genetic cross of a father with brown eyes and a mother with blue eyes. So you see, you might sometimes lose this mark here. It's fine, but you've already collected this one here. Then after writing those alleles based on the question here, you are going to write the gamete. You're going to write meiosis here. what's going to happen? The genotype is now going to do what? The law of segregation. Segregation is going to give you what? The gametes are going to be segregated. Then you write your gametes here. You separate them. From where? From the letters, meaning the letters that you had here previously. Let's say you had T, small t, you had small t, small t. You are just going to separate them to show that the law of segregation has taken place. And therefore, after writing meiosis, you write your gametes, because meiosis forms gametes. Remember, when you do the process of meiosis comes before gametes. Then those gametes, what are you going to do with them? You are going to fertilize. They are going to fertilize each other now. Then you do the process of fertilization. You can do it in a cross like this, or you can use a Punnett square. But this is fertilization. For writing meiosis and fertilization at the correct place, right? For writing meiosis and fertilization at the correct place, you are going to get 1 mark. Then you come and tell us what is now the genotype of the offspring. F1 stands for offspring. Right. For writing P1, telling us that the first type of genotype and phenotype that I was dealing with was that one of the parent. For writing P1 and F1 at the correct place, you are going to get another mark. So do you see that you can just write P1 and F1 down there, get a mark, write meiosis and fertilization, get a mark, and also write the phenotype and the phenotype of 2 parents that you are crossing. Then you get your free mark. That is how you get your 3 marks for free. You do not leave a genetic cross unsolved, right? So this is a format, always marked like this, meiosis, fertilization. Then you get your 1 mark. Doesn't matter if you get it wrong here, but those of you who are able to interpret, go ahead and get it right. So this is where they are using what? A Punnett square. Right, so we want to go next. So when you are doing a genetic cross, there are different types of inheritance, and you need to pay attention to the question to say which type of inheritance are they talking about. You will hear from when they are mentioning the offspring to say the offspring, all offspring had the same as the parent, then you will know that that one was dominant, or all offspring or offspring were different from the parents. So when you read your statement, look out for the type of inheritance they are talking about. We have different types of inheritance. We have complete, incomplete, co-dominance, and then this one, the sex-linked one. This is where you mostly make mistakes in the exam, because you think all genetic crosses are sex-linked. We will look at those ones that are sex-linked. Then a dihybrid cross, mostly you are asked to interpret the question thereof. Let us look at the complete dominance. Complete dominance. This is where only one parent's phenotype is evident in the offspring. Look, you are crossing a yellow pot with a green pot. But when you check all these offspring, they are all green. What does that tell you? It tells you that this is complete dominance, meaning that green is completely dominant over yellow. So this is an example of a complete dominance. So in your question, when they tell you that when you cross a flower pot that is yellow with the one that is green, all offspring were green, that information must tell you that they are talking about complete dominance, meaning green is completely dominant over yellow. Let us take a look at the second one. The second one is incomplete dominance. Incomplete dominance, this will be neither of the parents' phenotype is evident in the offspring. Let us take a look at our flowers. You are crossing a red flower with a white flower. But look at the offspring. All offspring are pink. Do they look like the red or the white? No, they have an intermediate color, and which is different from the colors of their parents. Now, this will tell you that this is incomplete dominance. There was no allele which was dominant over the other. But now, look at the letters. The letters are written in capital letters. Incomplete dominance can confuse you with the next one, codominance. Look at codominance. The letters, you have big letters still. But how will you know that they are talking about incomplete dominance or complete dominance? You are going to listen to the question. If the question says you cross a red flower with a white flower and all offspring were pink, then you know pink, it means different from both. If it is different from both, then it means it was incomplete dominance. Whereas with codominance, the statement will be the offspring had or was white with patches of red, or it was roan. Actually, a cow that is white with patches of red is said to be roan. You see, that is the key that will tell you what type of dominance are they talking about here, whether it's complete or incomplete or it's co. But the letters are also how they are big letters. So this is your codominance. Sex determination, you know how to read a karyotype. This is a karyotype of a male. Why is it a male? Because when you look at chromosome number 23, you see there is an X and a Y chromosome. A Y chromosome is small. Y chromosome is smaller than the X chromosome. So this is a karyotype that is showing you a male. And all other all other chromosomes from number 1 to number 22 are called autosomes. They are ordinary chromosomes. And then the chromosome number 23 are called gonosomes. They are sex chromosomes. Please pay attention to that terminology. Then this next one is a karyotype of a female. Why? Because at chromosome number 23, you see there are 2 X's, and all other chromosomes from 21 up to 22 are also still called what? The autosomes. Then how do you determine the sex? When you cross a male and a female, the chances are always 50-50. 50-50, because you are going to cross your X and your Y. This is your male with the X of the female. Remember, the female's genotype is always XX. So what are the chances of having a boy or a girl? They are always 50-50. X is XX, this is a girl. XY, it's XY. Then XX, XX, XX. Then X and Y, it's X and Y. So what are the chances there? It's 50-50. So always when a male and a female are crossed, there is a 50-50 chance of having a boy or a girl. So that is how you determine the sex. Now, the sex-linked disorders. This is where you are doing a genetic cross, where you are told that whatever or a particular condition is caused by a sex-linked, or it is sex-linked. So what are these sex-linked disorders? These are the genes that are located on the sex chromosomes. What are the sex chromosomes? They are the XX and the XY. So they are also called sex-linked genes. In humans, the term usually refers to X-linked. So even if you say they are X-linked, because they are mostly found on the X chromosome. Characteristics or genes located on the X chromosome. So you see, even if you don't say sex chromosome, you can use the word X-linked because they are found on the X chromosome. You can never find them on the Y chromosome. Why? Because the Y chromosome is small and it's short. So fathers can pass X-linked alleles to their daughters. Remember, if a child is a daughter or is a girl, it means the father gave that child his X chromosome. Then therefore, if his X was affected or it had a disease on it, or a sex-linked disease, that father is able to pass that disease to the daughter. Whereas mothers, they can pass this sex-linked allele to both sons and daughters. Because if a child is a boy, a mother can only give an X. And if she gives an X that is affected, then the father gives a Y, then you know that this boy child will have that particular disease. So this is an example of hemophilia, if the child can be hemophilia on the X chromosome. Right, so we want to go next. So this is just the inheritance. Then another example of sex-linked diseases is color blindness. The one that is in your syllabus is color blindness. So look at the examples. When they talk about color blindness, then you know you can use your X's to draw or to write. But if they don't say it's sex-linked, please do not use the X's and the Y's because it is not on the sex chromosomes. You can only just use your letters as plain as they are. Please, 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 you do not use X and Y if it is not sex linked. Read your question carefully. And if they want to give you something else that is not in your syllabus, they will tell you, they will give you a paragraph that is explaining that particular disease, and they will also give you or the information that it is sex linked. Therefore, you will be allowed to use the X and the Y. Blood groups. When you cross blood groups, they have their own specific alleles. So let us check. The inheritance of blood groups is an example of multiple alleles. So here there are multiple alleles controlling the blood groups. Now, the notation for alleles indicating blood groups is only A, I, A, I, B, and small letter I. So when you represent the alleles, you are going to use the capital letter I's and the superscript A or B or a recessive small I. I will show you how you use that. Now, different combinations of these alleles result in 4 blood groups. So these are just the letters that you use. Then when you combine them, they will give you 4 blood groups. So let us take the first combination. The first combination is this one. It shows you complete dominance. What is complete dominance? Look, for a person who is blood type A, we will take blood type A as a phenotype because that is something that is physical to them. Their genotypes or the alleles are going to be, it's either homozygous for blood group A, this is how you represent it, or completely dominant, like heterozygous for blood group A, where this big I with big A is going to be dominant over this recessive one. So this shows complete dominance. This person will have blood group A, but you can indicate it with, if they say it's homozygous blood group A, then you use big letter I with big letters A superscript, big letter I with big letter A superscript. So you must pay attention to how you are going to write them. Then the second one, for a person who's blood group B, the same applies. A person who is, the statement will tell you homozygous blood group B, you are going to represent it in this way. Heterozygous for blood group B, you are going to represent a dominant one and the recessive one. So that is why it is very important that you highlight or you listen to the question, what they are saying or what is the blood group before you cross. Because if you get the genotype wrong, you are going to get the answers incorrect as well. Then for a person whose blood group AB, this is co-dominance, meaning there is no allele that is dominant over the other. They are both expressed in the genes, or the genes have both blood group A and B as completely dominant. So they are co-dominant. And then for a person whose blood group O, they are recessive. Now recessive next is complete dominance. Now incomplete dominance. Now this one is incomplete dominance. There's no one that is complete over the other. So this is how you are going to show the alleles for a person who has blood group O. So when you cross, when you do a genetic cross with blood groups, remember you are using these letters for the alphabet. Remember homozygous, remember heterozygous. Right. Next, then you have your genetic pedigrees. Genetic pedigrees, this is where you must get a lot of marks. You must be able to interpret a genetic pedigree, because a genetic pedigree is the one that traces inheritance of characteristics over many generations. So you must be able to tell us that this was the first generation, second generation, then be able to tell us which one is the males, which one is the females, which ones are affected, which ones are not affected by the disease. So be able to read that information. But how do you solve a pedigree diagram? So these are the things that you need to learn. The circles represent the females, the squares represent the male, unless told otherwise. Then the shaded block will represent the affected trait. Then the one that is scratched like this will represent the deceased. I know many of you maybe have not come across this one, but please take note of that one. Then the one that is joining like this shows you these are the twins, and then the one that is in brackets or square brackets like this shows you that these are adopted. Then the triangle will show you that these are the kids that were miscarried. Then you must understand that the line between or going across horizontally shows that there's a marriage. The line that is going down shows the descendants, that these are the children coming from the previous marriage. And then the line joining here shows you it is not joining in the middle. Do you see? It is showing the sibling line. And here you see the first row will represent the first generation, and the second row has your second generation. So this is the information that you need to know. How do you solve a pedigree? Study the key. First thing, what are you given here? You read that, because it will give you information. Look for the dominant and the recessive characteristics. While you are reading, look at what are they saying is the dominant one, what is the recessive one. Fill in the genotype of the individuals with the recessive condition. When you see that this one has the condition, go and fill in all the small letters. Then from the small letters, you know that each individual with the recessive condition inherited one allele from each parent, meaning you are now going to do what? Work backwards and fill in one recessive allele for each parent. If a child has AA, then you know that for them to have this AA, each parent must have had a small a. You see, now you go back and you fill in those small a's for the parents. And then if the parent has a dominant characteristic, fill in the capital letter and the second letter. So you will put in maybe your big A there, because now you see the parent is not affected. It has the dominant characteristic, depending on whether that particular disease is controlled by a dominant or by a recessive gene. Then any other individual showing dominant characteristics is mostly homozygous dominant. Then you can just complete filling in those with two capital letters. You work backwards after doing these first three bullets there. Then paternity testing with blood groups. You know that we cannot determine the paternity just using blood groups, because there are only 4 blood groups. So the possibilities are not as many. So you cannot ascertain that this child belongs to this father, because even the father next door can have blood group O. So there are only 4 possible blood types. So you cannot determine paternity using blood groups. It's not very accurate. However, DNA profiling can be used to prove paternity, because now then the sequence of the nitrogenous bases will be compared, and when they are compared, they can match. If they match, then you know that for certain that the father, this person can be the father, or this person can be the father of a particular child, by just comparing what? The sequence of the nitrogenous bases there. I think we can leave it here. We have done 2 of our most biggest topics for paper 2. So you are already at about 100 marks. So please make sure that you go and you complete the other topics, that is your DNA and meiosis. Those are smaller topics in paper 2. You have the whole weekend to study for this paper 2. I hope you're going to do well. Go and do your previous question papers. Look at all the tips and the videos that we have done up to this far for revision, and answer the questions that can help you to be ready for the exam. I would like to thank you so much for watching this lesson for today, and I wish you nothing but the best for your paper 2 on